All right, listen up. Let's go through the cell cycle. And I want to go to an interesting story. This is going to be the story about human papillomavirus. Now, I'm guessing that about half of the class may have already been vaccinated against this. This is called HPV, and it's a vaccine that you would normally get somewhere around 10 to 12 years old, is right around the range where they would normally get it. And it protects against cervical cancer. And it's able to do this because the cervical cancer is actually going to be caused by a virus, human papillomavirus. And if you're immune to the virus, you are therefore immune to cancer. It's one of the few kinds of cancer we can actually prevent, which is really neat, I think. Okay, but first of all, we got to get started with our story. And then on the last slide of this set, I have diagrammed both the test question. I may tweak it a tiny bit to make the wording a little more clear, but that's going to be the question. And I've laid out a reasonable path to an answer. So I'm not going to put this up as a whiteboard episode, but yeah, boy, have I left a whole lot of breadcrumbs for you to fill in. So, yeah, you may have to click back through 4 or 5 slides. And if you run into words on these slides that you're saying, wow, this doesn't completely make sense, I would say the thing to do is to get out and look that word up. And you can look these up on Wikipedia. That's a very good place. There are lovely pages on all of this stuff because it's fairly basic biology, and you will have learned something, and you will have also learned how to dig yourself out once you run into problems. And believe me, that is something that happens over and over and over again, and will continue to happen in your career. And yeah, as long as you're good at digging your way out, that is a very valuable skill, and you will become a useful person in whatever field you end up in. Okay, so cell cycle. Cell's going to go through this particular cycle, and at the end of the cycle, the cell is going to divide, and then the rest of the cycle is basically getting ready for division. Now, if all of your cells just kept dividing indefinitely throughout your entire lifetime, one of two things would happen. Either you would grow continuously throughout your lifetime. Can you imagine? Like you get, little kids that grow up, and when they get into their teens, then they kind of stop growing. But can you imagine if people continued growing? Your grandmother at 70 years old, or however old she is, would be massive. She'd be like 5 times as tall as your typical teenager. She'd be dunking basketballs and just barely able to stand under all of her own weight, probably. That'd be a weird world, right? The other possibility is that if just some cells grew indefinitely and other cells did not, and there was kind of a breakdown in this process, you would have uncontrolled cell growth, which is what we know as cancer. And when basically one part of your body is growing faster than it should be, that's what cancer is. And yeah, it doesn't benefit the host in any way. It's just some cells that have kind of broken off on their own and decided to go it alone. So here we go. Let's talk about the phase. So you got your interphase, and this is most of the life of the cell, roughly 90% of the cell cycle. And it's going to be broken up into the gap phases. Gap 1, followed by S phase, with its synthesis, where we're actually making another copy of all of our DNA, copying our chromosomes. Then G2 phase, which is the second gap phase or the second growth phase. And during G1 and G2, the cell's going to store up some of the raw material that it needs to go through the S phase or the later phase, as depending on which stage we're talking about. All right. And so the cell can grow, it can accumulate energy, it can use energy during all these phases, but it only makes its chromosomes during the S phase. So, quick quiz. If I looked in a cell, when would I find the most DNA in that cell? And it would actually be about twice as much as you would find at other times. Have you got the answer? It's going to be in G2 phase, right? So G2 phase is after S phase. During S phase, we're going to be making that second set of DNA. And once we've made that one, then we are effectively at the G2 plateau. We're going to sit in G2 phase until we can undergo cell division or not. Yeah, some cells never undergo that division. They'll get to a certain point and they will stay there. And so there are these checkpoints. And so that's the little red boxes you can see in the diagram at lower right. And these checkpoints are where the cell cycle is stuck. It can go up to that stage and then it will just stay indefinitely at the G1, G2, or M checkpoint until and unless just the right combination of genes are expressed that will drive it past the checkpoint and on around the next cycle. So if you look, these checkpoints are in interesting places. So there's a checkpoint in G1, but it's not actually at the end of G1. So we don't just start synthesizing DNA, as soon as we go past the checkpoint. The checkpoint is actually during G1, and the cell will sit there for a while, accumulating enough DNA to actually be able to copy all of its own chromosomes before it actually starts the copying process. There's a lot of getting ready that you have to do before you can actually duplicate all the chromosomes inside of a cell. So that kind of makes sense. Then there is a really strong checkpoint at the end of G2. And so the phase after G2 is going to be the mitotic phase, or M phase. This is where the cell is going to divide. And so your anaphase, telophase, all these things happen during M phase, which is together just a little tiny sliver. And we're going to assume that that stuff basically just happens. There's a checkpoint in the middle of the M phase, which I believe is at the point where you are pairing up your chromosomes. So you have a little checkpoint to try and make sure you've paired everything up, and you're not going to end up with a cell which has too many chromosomes and another cell which is maybe missing a chromosome, because that would be bad. Yeah, usually for both cells, that's very bad. Yeah, all right. The G2 to M checkpoint, this is very, very strong. And cells will quite often get stuck at G2 and not be able to make it through. However, if you are thinking not as a cell, but as something that's trying to use a cell, like, for example, human papilloma virus, what you want is for the cell to not have any checkpoints. You want for this cell to keep going around and around and around, because as the cell goes around, it keeps stacking up raw materials like nucleotides to make RNA or DNA, and lots of ATP to drive the entire process. It takes a lot of ATP to go through each of these cell cycle stages. And so from the virus's point of view, you would rather that there were not any checkpoints. But there are checkpoints, and so to be a good virus, what you have to do is you have to find a way to knock down the checkpoints, basically blow up the checkpoints, so that the cell will keep on dividing, which means you can keep on growing and make a few extra little viruses a little bit faster. It's a competitive advantage, and viruses are real good at this. Almost every virus out there is doing something to your cell cycle and usually trying to drive your cell cycle a little bit faster. Yeah, the cells fight back, but viruses are pretty good at their job as well. So this is where we're going to be going with this lecture. Now, as for the consequence of this, yes, it does help the virus a little bit if it can knock down those checkpoints, but what does it do to the cell? Oftentimes, when a virus knocks out a checkpoint, it's actually going to knock out that checkpoint permanently. And that means that this cell will no longer have a checkpoint, which means that the cell can just continue to grow as if there was no checkpoint there. And a cell that just continues to grow with no checkpoints is called cancer. So, the virus gets a few extra copies of itself out of this. It can infect a little bit further and faster. But for the host is going to be left with cancer. Yeah. And if you think that's a bad deal, yeah, I totally agree with you. That is a very bad deal. For the host, it's a great deal for the virus. And frankly, that's all the virus cares about, I would say. Okay, so how do we drive the process around? What are these blocks? Well, there are a set of keys that will open up each of these blocks in the cell cycle. And these keys have two parts. They are a cyclin. So this is a protein that is going to be able to unlock another protein, which is called a cyclin-dependent kinase. So the cyclin is like the key, the cyclin-dependent kinase is like the car, and the car is going to drive around smashing through roadblocks, Grand Theft Auto style, I guess. Yeah, that's basically how it works. So, cyclin-dependent kinases are also proteins, and a kinase is a protein that's going to take a nucleotide like ATP. And I think all these cyclin-dependent kinases do use ATP, but there are other kinases that will use GTP or CTP, something like that, which is fine. You just need it as a source of phosphate. So what a kinase does is it's going to break the third phosphate off of an ATP, so it's going to turn it into an ADP molecule, and it's going to take that third phosphate and attach it to some other protein. Now, a phosphate attached to a protein is usually going to be like a little switch. Most commonly, it is an on switch. Occasionally, it is an off switch. But for the purpose of this, let's just assume it's an on switch. And so when we make the cyclin, it will activate the cyclin-dependent kinase that's always just waiting around, like a car that's just sitting there, waiting to be hot-wired and driven around. And once you've made the cyclin, that is the key, and that controls the cyclin-dependent kinase, and that will then drive us right through one of those cell cycle roadblocks. So there we go. All right, so why do all cells not just, drive right past the roadblock every single time? Why is there cell cycle arrest? It's because of this guy, P53. Think of P53 like the police. Yeah. In this case, the police are saying, don't drive the cell cycle so fast. Let's wait. Let's wait until it's the right time. You got to follow the rules. That's what P53 is all about. It's about following the rules. So the reason you have a P53 is that it is going to be turned on when there is danger in the form of DNA damage. And just walking outside on even a cloudy day, you're going to have thousands to hundreds of thousands of DNA damage events happening in every single cell on the surface of your body. And this is going to happen within minutes of walking outside. You're going to pick up massive DNA damage. The only reason why you are not a walking ball of cancer right now is because you got P53. And P53 says, whoa there, cell that just got DNA damage. Let's wait a minute before we divide. Because if a cell tries to divide with its DNA damaged, it's going to pass that damaged DNA to now 2 cells. And those are going to divide and pass it to 4 cells. And damage is not good for these cells. Damage can lead to cancer. Damage can lead to all kinds of bad stuff. When P53 gets damaged, when you basically don't have this little policeman running around your cell saying, whoa, wait till we repair the damage before we divide, that's when you get cells that are dividing uncontrollably, and that is a very common condition that leads to cancer. A lot of skin cancers have mutated P53, and if it gets damaged, then there's nobody to tell your cell, hey, don't divide. Don't just keep dividing forever. So P53 is kind of indirectly one of the cell cycle blocks. Yeah. Okay, so how does P53 work? Well, P53 is going to activate a protein called P21. And so think of P21 as drinking age. So you think of this as a little policeman there saying, hey, yeah, P21, yeah, you got to show me your license. And if you're over 21, then it's fine. And if you're under 21, you're in trouble. P21's job is to actually inhibit cyclin E. And cyclin E is the one that is going to run cyclin-dependent kinase number 2. Cyclin E and cyclin-dependent kinase number 2 are a really powerful combination. These two can drive the cell past that G2 cell cycle block. When there's a lot of P21, when there's activated P53, you make a lot of P21. That P21 puts a giant inhibition on cyclin E and cyclin-dependent kinase 2, and therefore the cell does not grow. The cell cycle is arrested. And arrest, is the term that biologists actually use, and so I think it works with the little police metaphor, if that makes sense for you. It doesn't matter how you remember these things. It just matters that you can build them into some kind of a story so that when you think of one part, you can think of all the parts. Like stories we tell around, the night before Christmas or whatever. You get that one little part, 'twas the night before Christmas, and then you can just keep going and fill in the rest of it. That's the goal here. We want to build a stupid little story in your mind that's actually going to stick with you better than the individual parts would. And by doing this, by building these little stories, eventually you're going to find out that one of these stories that you've built is going to bump into some other story. Maybe in cell biology class, maybe in immunology class, maybe in virology class, down the road, maybe in microbiology class. But if you get enough of these stories, you've made a little map of a piece of the world, and eventually it's going to connect up with some of the other pieces. And then once you've got those connected, boom, that area is yours. You occupy that area, you own that area, you know that area. So that's the goal. That's why I'm teaching this in the way that I am. So the alternative is, if we have low amounts of P53, so P53 is not being activated, then we are not going to make very much P21. And so P21 is not going to do a good job of stopping cyclin E. And cyclin E is just going to get into its cyclin-dependent kinase number 2 and tear off around town. And yeah, it's going to boom, smash through that cell cycle arrest. And we call this escape. So think of it as like a little escaped prisoner, cyclin E. You gotta keep that cyclin E locked up unless it's time for the cell to divide. But even then, it's got to be done carefully under the right conditions. All right, so this is the situation. P53 is there to make P21. P21 is there to shut down cyclin E. And if you got no cyclin E, then your cyclin-dependent kinase 2 is not going to be activated. And if you got no cyclin-dependent kinase 2, then you are not going to divide that cell today. No sir, no ma'am. Now, from the virus's point of view, this is all just terrible, right? This is the worst. So a virus like human papillomavirus needs to be in dividing cells. It's actually depending on dividing cells for its very growth. So when the cell divides, it does two different things. One, it makes a whole bunch of resources that human papillomavirus can use. That's great. The virus likes this. The virus would say thank you if it could, but viruses don't really say thank you. The other thing it does is that this cell, once the cells that have the virus in them start to divide, the virus actually gets copied for free. And so each cell that divides is now going to come preloaded with a copy of the virus in it. And each of those cells is then going to continue dividing. And so you're going to get a nice little tumor in there, or not very nice, because this can be any number of cancers. So the way this works is that our virus can't really do anything in the outer layers of cells. The outermost cells on your epithelium are actually either dead or almost dead throughout your body. Humans are basically dead on the outside. Yeah. Yeah. But it's living stuff on the inside. Yeah. And so what the virus needs is it needs to get into the lower levels of cells, because the cells are going to divide from what is called a basal level. And the basal cells are going to divide up here and become the spinous cells. The spinous cells are going to compact and become the granular cells, and the granular cells are going to basically just dehydrate. And they're going to become the cornified cells, and those are actually the outer layer of the epithelium. So the only way for the virus to get in is with friction. You need something to be rubbing on those cells to try to basically wear away some of the cells or create a little tiny gap where this virus can go down and get to those basal cells, because it can't grow unless it can get into the basal cells. This is why HPV is a sexually transmitted disease. It's actually the rubbing during sex which is going to create the little gaps, the little openings that are going to allow HPV to get into one of those layers. And so if you have plantar's warts on the bottom of your feet, this is because you were walking around on, in my case, the YMCA floor where somebody else with one version of HPV had walked before, and you had just enough friction on your feet to give a little gap so that the virus could get down to that basal layer and start to grow. If you've got a little skin tag on your eye, like those little skin lumps, like a little tiny dehydrated wart, yep, that is also human papillomavirus. Actually, warts pretty much anywhere on your body are human papillomavirus, and they only happen when you have enough friction on that part of the body. So from like rubbing your eyes, when you're a little bit sleepy, if you happen to have a little human papillomavirus on your fingers when you rub your eyes, then yeah, that's how you're going to end up with the little skin tag there. And it's fine. These are cancers. The virus is causing cancer when it causes a wart. That's what a wart is. But for the most part, when the warts are in your regular old epithelium, like on the outside of your body, usually doesn't spread, and these are usually what we call benign cancers. So it's like a little tumor, but it's not a bad tumor. So pretty much anybody that is not a tiny newborn baby is going to have somewhere on their body some at least little tiny bit of human papillomavirus cancer. And this is not something that is good for you. It's just something that kind of happens as you move around the world and run into other people and are in the same environment as other people. It's kind of an unavoidable consequence of living. At least it was unavoidable, yeah. Until we made human papillomavirus vaccines. And they actually block the virus from being able to replicate. You will actually make a good immune response, shut this virus down, and it will prevent you from getting some of these papillomavirus tumors. The tumors that the papillomavirus makes in the throat or the penis or on the cervix, those can be very serious, and they will often spread throughout the body. And those can be very aggressive tumors, and those can lead to death. They can be fatal tumors. And so protecting against human papillomavirus is, really important in protecting against cancer. This is the one totally preventable kind of cancer. Now, the problem is that there are about 300 kinds of human papillomavirus out there that we know about, and they're finding new ones all the time. The vaccine protects against the 4 that are associated with the most cases of cervical cancer. So it's kind of targeted at those. You could make other vaccines that would protect against all the ones that cause other kinds of warts. That's totally doable. It's just that, we're tackling the big problems first, and then we're going to work our way down the list. I think that's the idea. So anyway, this is what the virus is after. It's looking for rapidly dividing cells that come preloaded with a copy of human papillomavirus. And so it is going to go to great lengths to make sure that those cells continue to divide, and so that there is going to be more virus in the world, because that's what viruses want. They want there to be more virus around. Okay, how does human papillomavirus short circuit our actually very good system? How does it defeat the police of the cell, P53? Well, the answer is it's got a little protein, and human papillomavirus actually has several ways to defeat the cell cycle and blow past all those cell cycle arrests. We're going to focus on one because this is not virology class. We're using this to illustrate a point about biology, about cell cycle biology and cancer, basically. And this is infectious cancer. So it's, interesting case. And one that will affect almost everybody who is listening to this lecture at some point in their lives. So, human papillomavirus makes a protein, and this one is going to be called the E6 protein. And it's an E because it's an early gene. It's made fairly early after the virus after the virus actually infects the cell. And E6 is going to be one of the genes that is responsible for causing cervical cancer. If you're able to knock out the E6 gene, the virus still has a couple others that will probably cause cancer, but this is the most powerful one. And this is the one that interacts directly with P53, and so that's why we're going to talk about this one. It's connected to the little cell cycle protein that we are studying here. So, E6 binds to a protein that's called the E6 associated protein. So you can see it over here. It's the E6AP, E6 associated protein. And it was named by somebody who found it after they had discovered E6 and were trying to figure out, well, how does this little tiny E6 protein, which is just a little tiny bit of a thing, how does this actually cause cancer? Because it seems like really little to have such a big effect. And the answer is, it has actually a very little effect, but it's a powerful little effect. So E6 is going to bind to the E6 associated protein, which is a protein that's already there in your cells. And it probably would have had another name, except that people discovered it after they discovered E6, and that's the one thing they knew about it, and so, that's its name. The E6 associated protein is a thing called a ubiquitin ligase. And it takes 3 steps to make a ubiquitin ligase. Now, rather confusingly, they're called the E1, E2, and E3 steps. Since we're talking about the E6 protein, which is a human papillomavirus protein, I'm not going to mention anymore that there are E1, E2, and E3 ubiquitin ligases. We're just going to call this the ubiquitin system. And ubiquitin is a little protein, and it's shown as little dots up here. And this protein can be covalently attached to other proteins. Lysine amino acids on proteins are a good point where we can attach the ubiquitins, and yeah, they'll get attached onto there. Then you'll have a protein that has ubiquitin bonded to it. So yeah, the ubiquitin acts as a little molecule. It's kind of like a little tag. And the ubiquitin will often change the function of the molecule. So ubiquitins can mark a molecule as damaged. They can mark a molecule as activated. Or a common thing is that they can mark a molecule that is old and needs to be degraded. So this is a way that we can get rid of proteins. We attach a ubiquitin to them, and it's like a great big kick-me sign, and that protein will get led off to the proteasome, which is like a great big garbage shredder, and the protein will just get shredded. The ubiquitin will get recycled, and it'll go get attached to some other protein. And when we're going to mark a protein for shredding, we don't just add one ubiquitin, we add a big long chain of them. And so you can see a big branchy chain of ubiquitins over here. And the exact branching on that chain, the exact number of ubiquitins, all of this has meaning inside of the cell, and all of this kind of changes the way that the ubiquitin is going to function. So ubiquitin is kind of a complex molecule, but the most common way that ubiquitin gets used, or the commonest thing that we are going to talk about anyway, is ubiquitin being a marker for sending proteins to be degraded and recycled. Just broken down into amino acids that we will then reuse to make some other protein. Okay, so E6 acts like a little bridge. It binds to the E6 associated protein on one side, and on the other side it binds to P53. Ordinarily, the E6 associated protein would not come anywhere near P53. These two things might bounce off each other somewhere in the cell, but they would not be stuck together for any length of time. But because the E6 protein is like a little bridge, it's sticking them together, now the E6 associated protein does what it does, which is it's going to call on these other proteins that will add a ubiquitin molecule to the protein it's attached to, which in this case is going to be P53. And it's going to not just add one, but it's going to add a whole chain of them. And so here's our whole chain of ubiquitin molecules on P53. And once these molecules are added, then you know what happens next. P53 is going to get broken down, recycled, and shredded. Now, ordinarily, if this is a regular protein, that's fine. Proteins get old, they get recycled, we make new proteins. Yeah, we might even make the same protein again. This is just part of normal cell business. When the cell is growing, when the cell is living, it's constantly breaking down and making proteins. That's just part of the way that a cell works. But when P53 is gone, there's no policeman. So there's no one to make P21. And if there's no P21, there is nothing to stop cyclin from getting into that cyclin-dependent kinase 2. And so our cyclin E is going to operate cyclin-dependent kinase 2, and it's going to run all over the cell, adding phosphates onto different molecules, breaking down ATP, and causing that cell to go from G2 phase into M phase. And once it's in M phase, it's going to go through that M phase block, and it's going to replicate. So this virus, by making E6, is going to force the cell to go through a cycle of division. And once E6 is made, it's real stable. So it's going to stay attached to the E6 associated protein. And anytime the cell tries to stop by making P53, E6 associated protein is going to just put ubiquitin all over that P53, and it's going to be shredded every single time. So every time you try to make new police for your cell, boom, they're gone. Yeah, they are defunded, if you like. And so, yeah, no P53, no cell cycle arrest, no P21. Cell goes crazy, keeps dividing, and the virus wins, and the host gets cancer. Yeah. So the host loses kind of twice. It's got the virus in it, which is growing very well, and the host is probably going to get cancer of some sort. And you just hope at that point that it is a benign tumor and, yeah, not a metastatic one. Okay. And so, as a special added service on this lecture, since this is a new one that we're putting in this year, in place of what was, frankly, a rather uninspiring old cell cycle lecture, now it kind of has a point. And I like lectures with a point a little bit better, a little bit of real world relevance. So our associated test question is going to look something like this. I may tweak it a tiny bit, but it's going to be explain how cyclin E, you remember that? That's the one that operates cyclin-dependent kinase 2. So how's this normally blocked from driving cells to divide? Well, the answer is that cyclin E is normally not there. We only make cyclin E when there's no P21 around, because P21 is normally stopping us from making cyclin E. And when cyclin E is there, it's going to operate cyclin-dependent kinase number 2, and that is then going to go off and do 50 other things, and we don't have to name the things, but that is going to cause the cell to divide. And that's its own whole lecture right there. And frankly, you can have that lecture in cell biology class when you're a junior. For now, this is a little taster that'll get you into it. So, okay, that's kind of the first part. And how is human papillomavirus able to overcome this block? Yeah, the fact that there's no cyclin E around. How does HPV do it? Include in your answer how rapid cell division affects the virus. It really helps the virus, of course. And how it affects the human host. Gives the human host cancer. And the best you can hope for at that point is that the cancer is benign. Yeah, so that's no good. So if you were breaking this down, what are the steps you would need to fill in to make a nice, complete answer? Here is a rough roadmap. And so you're going to have to click back a couple of slides or get out your notes and fill in all of these parts. And then once you've got that, you've got yourself a really good model answer that you can study for the test and, yeah, use on the test to do really well. So, yeah, study it, learn it, pack it in your brain, get it all loaded up, and then it's yours. And it'll be yours for this class and for life. This is interesting stuff. I'm going to make you a little bit more interesting around the Thanksgiving table. I want the conversation to revolve around you and your new science knowledge. That is my goal for this class for you. So take that for what it is. Okay, so first part of a good answer, you're going to start with the cell cycle. You're going to talk about, we got our G0 phase when we're just not going to divide at all. We got our G1 and G2 phase where we're static, but we're building up towards something. We got our S phase, and we got blocks. We got a block at the end of G1, we got a block at the end of G2, and we got a little block which is actually fairly easy to overcome that's right in the middle of M phase. And 90% of the time we're going to be stuck in the interphase, which is G1, S, and G2 phase, and the other 10% of the time we are going to be dividing. Yeah, okay, so what's a cyclin? What's a cyclin-dependent kinase? Oh my gosh, we have a slide on that. I even put a picture of cyclin bound to a cyclin-dependent kinase in there. Remember what cyclins do. They're going to activate cyclin-dependent kinase. Remember what a kinase does. It's going to break ATP and put a phosphate on other proteins. And in this case, that's as far as we need to know. But if you want to look it up, what some of the targets of cyclin-dependent kinase 2 are, these are going to be some of the proteins that are involved in cell division. This is the block on the cell cycle is not just one factor. It's a whole bunch of things that have to happen. But cyclin-dependent kinases are kind of the thing that activates all of those things and starts the process going. So what's P53? Remember our little policeman? Yeah. How does P53 block cell division and why? Remember that P53 is active or is activated and made in response to DNA damage, among other things. But yeah, when your DNA gets damaged, so that we don't replicate a cell that has broken DNA, we just tell the cell, wait. And P53 is the helpful little policeman that's going to tell that cell, wait a minute, let's repair this, and then you can go on and you can divide, and it'll be fine. And so how does P53 work? Why, it's going to work through P21, and P21 is going to inhibit production of cyclin E, and with no cyclin E, you're not going to be able to operate your cyclin-dependent kinase 2, and so the cell is just going to sit there in G2 phase, just looking at you. Okay, what's E6? Why, it's a human papillomavirus protein called E6, made from the E6 gene. How's it get rid of P53? Well, that's a story in itself. So you're going to mention the E6 associated protein. You're going to mention the ubiquitin ligase system, and ligase is Ligase. The ubiquitin ligase system is going to put ubiquitin molecules onto p53. We're going to add them onto, it's actually lysine 48, K48 linkage of ubiquitin. You don't have to know that, but you're going to run into it if you end up in immunology or virology someday. Probably if you're going to end up in cell biology. So, whatever. Tuck it away in the back of your brain, and it's yours. It's a little gift, like a little mint they give you when you're leaving the restaurant. Okay, ubiquitinated P53 is going to then get shredded, and if there's no P53, there's not going to be any P21, and if there's no P21, then there's nobody to tell the cell not to make cyclin E, and so the cell's just going to make cyclin E, because that's what it does. And when the cell has made cyclin E, then the cyclin E is going to operate the cyclin-dependent kinase number 2. Cyclin-dependent kinase number 2 is going to go around phosphorylating a bunch of stuff, and bam, you got yourself a cell division event occurring. How does that benefit the virus? You know how it benefits the virus. The virus gets free copies made of itself because it's inside the cell that's dividing, and the virus gets to live in a cell that is storing up vast quantities of nucleotides that the virus can then use to make more viruses. The cell was going to do something with that, it put its name on that and put it in the fridge because it wanted to use these nucleotides to make more cells, but the virus comes in like a terrible office mate, raids the fridge, and turns it into more viruses. Viruses, they are just the worst, aren't they? Okay, how does the uncontrolled cell division affect the host? Well, it gives the host cancer. That's basically the long and the short of it. And that cancer can then spread because this E6 can be a permanent feature. Often one of the things the virus will do is it will actually insert the E6 gene into the genome of the cell so that wherever that cell goes and whatever children that cell makes, they're all going to come preloaded with E6, which is going to shut down P53, which is going to make that cell divide uncontrollably now and forever until that cell takes over the entire body, replaces organs, disrupts neural pathways, and basically kills you. Yeah. So that's why human papillomavirus is bad. Stay in school, kids. Don't go get human papillomavirus. And that's what P53 is there for. This is how the cell cycle works. So I hope you like that. I thought it was a nice, concrete example so that we're not just learning anaphase, telophase, whatever, interphase, prophase, blah, 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 so that we're actually doing something real that's connected to human health and to something that probably is affecting someone around you right now. So there we go. That's the end of our lecture. Study up, use this, get a good grade on the test. You'll make me happy. You'll make yourself happy. We're all happy. Yeah, do a good job. Thanks a lot.